Hi everyone, in this video we're going to look into theories that explain the origins of life and cells. In our previous video we focused on the protocell first theory, which kind of argues that a protocell, which is a simple cell-like structure formed from lipids or fatty acid membranes, would have kind of aggregated into this vesicle-like structure when it was exposed to water, and thus formed the primitive cell, the prototype of a cell, which would have later on gone on to adapt functions like metabolism, replication, etc. to become the cell and life as we know today. Well, in this video we're going to focus on different theories. We have the gene first theory and the metabolism first theory as the two other most likely theories that would explain the origin of cells, but in this video we're going to focus more likely on the gene first theory. Now, the gene first theory really fundamentally grounds itself in the idea that life began with molecules that are capable of storing information and self-replication. Essentially, it's saying that life began with genes. But genes here is not defined only as modern DNA, it's also defined as generally an umbrella term as molecules capable of storing and passing on information. And as of today, the most likely candidate for the gene explained in the gene first theory is RNA. And this is because RNA has both functions of storage, it's capable of storing genetic information much like DNA, but at the same time it has catalytic properties, which is characteristic of enzymes and proteins. Therefore, because the gene or the RNA is capable of doing both of these functions, it's most likely pointed out as the likely gene in the gene first theory. And the gene first theory states that instead of metabolism or cells coming first, information storage and replication came first, functions of the gene. And the gene first theory also grounds itself in the Darwinian evolution logic, which states that there are three components necessary for evolution of cells: heredity, variation, and selection. And these are all displayed by the gene in the gene first theory. Heredity, in the sense that replication will copy the sequence of the gene, copy the base sequence, nucleotide sequence, and this is going to lead to heredity. Variation, in the sense that mutations can happen during the replication process. And selection, in the sense that these variations will arise and better replicators will arrive, and they will also survive and dominate. So this idea of natural selection. All three components are present with the gene discussed in the gene first theory, thus it fits the criteria of Darwinian evolution, which is necessary to explain the origins of cells and how these origins of cells became the cells that we know today in our world. Now, we normally just generally point out the RNA as the gene referred to in the gene first theory, and there are a couple of reasons for this. First reason is because RNA is capable of storing genetic information. We all know that RNA is a polymer that's made of four types of nucleotides, which is A, adenine, U, uracil, and then G, guanine, and C, cytosine. And it's essentially a linear sequence of nucleotides that can essentially encode information, just pretty much like DNA. And each RNA molecule can be copied on to produce identical sequences, and this ensures the factor of heredity. Also, it allows for replication in the sense that A pairs with G, and A pairs with U, and then G pairs with C, and this will allow RNA to essentially act as a template of replication, and thus replication is secured. Also, mutations can arise, due to errors during copying processes, during substitutions, deletions, or even insertions of certain base sequences, and this is going to lead to variation. And it also fits the Darwinian sort of evolution theory in that sense. Now, one thing that makes it very different from DNA is that it is generally less chemically stable, and therefore it's a little bit more reactive than DNA, and therefore it's kind of a disadvantage in the sense that DNA would make a better, safer and more stable storage medium. But still, we know that the RNA is going to be a replicable and mutable code, and thus it's going to fulfill the role of the gene discussed in the gene first theory. Second is that the RNA is capable of catalyzing reactions, and this is pretty much the functions of proteins acting as enzymes and catalysts. RNA doesn't necessarily exist only in a linear chain format, but it can also fold into secondary and tertiary structures. And we know that these specific specific molecules called ribozymes is going to act as catalysts. It's going to have catalytic functions, and such as RNA splicing or even polymerization, and we know that therefore RNA is going to display some catalytic properties to an extent. Now, this is very important because the RNA is now able to store information, and at the same time it's going to perform the chemistry that's absolutely necessary to propagate itself. proteins can't do that because it doesn't necessarily have a templated inheritance, and DNA also can't do this because it doesn't really have a catalytic ability. So we see the RNA as something that can do both of these properties that's necessary for sustaining life, essentially. Third evidence for why RNA would most likely be the gene discussed in the gene first theory is that it's found in a lot of organelles and a lot of modern biology processes today. For instance, we know that ribosomes, they're essentially just like RNA machines. They have ribosomal RNA. So we see RNA also again in transfer RNA, also known as tRNA, messenger RNA, mRNA, and there's a lot of RNA viruses as well today, which all goes on to show that RNA is found a lot in modern biology today, and it also kind of acts as this verification for the RNA world theory. Speaking of which, the RNA world hypothesis kind of states that because the RNA was capable of doing both functions of DNA, storing genetic information, and also catalytic properties of a protein and an enzyme, it would have kind of been the intermediary or the predecessor stage, which came before the DNA and the protein world. Now, the DNA in the protein world is pretty much just like the world like of today, biology today, in that DNA, which is a much more stable genetic storage storage molecule than RNA, substituted the function of RNA as storage molecules. Protein, which is a much more effective catalyst, really substituted RNA. But essentially, the RNA world hypothesis is that RNA molecules kind of acted as this intermediary stage prior to the coming of the DNA in the protein world. And alright, that is pretty much it for the gene first theory. In our next video, we'll be looking into the other theory, which is the metabolism first theory.